Today we'll be investigating the movement of mollusks, specifically gastropods, by summarizing the study Muscular waves contribute to gliding rate in the freshwater gastropod Lymnaea Stagnalis. Snails, or gastropods, fall under domain Eukarya, kingdom Animalia, and clades Bilateria, Protostomia, Lophotrochozoa, and Mollusca, which is then categorized by the presence of a head, visceral mass, and foot. Gastropods can be differentiated from other mollusks by the torsion present in their coiled shells. The crawling mechanism used by gastropods depends on whether their primary environment is terrestrial or aquatic. Land-dwelling gastropods creep via a series of smooth muscle waves, while their aquatic counterparts gallop or glide using cilia.
The great pond snail, L. stagnalis, uses a combination of overlying layers of smooth muscle cells and ciliated epithelium for locomotion. In aquatic gastropod, the organism glides gradually by means of ciliary beating. Many mollusks, such as L. stagnalis or H. aspera, a common garden snail, have large muscle cells distinguished by their giant mitochondria that line the horizontal muscular system. This shared trait suggests a similarity in reaction to various stimuli. Based on various morphological traits shared with H. aspera, it was suggested that L. stagnalis has both ciliated and muscle cells that contribute to overall locomotion. The ciliated cells powered by glycolysis and the large mitochondrial cells powered by respiration. Previous studies observed that inhibiting respiration in gastropods resulted in a halt to movement.
In this research study, scientists conducted numerous trials to determine the effects of serotonin, both alone and in the presence of cyanide, on the locomotion of the L. stagnalis, or great pond snail. Each of these trials were conducted in an anaerobic environment as well as an aerobic environment, in order to discern which muscle cells were the primary influencer of locomotion, since cilia function regardless of the presence of oxygen. In order to determine the effects of serotonin, or 5-HT, and cyanide, or KCN, on the locomotor activity, scientists pinned soles of the L. stagnalis great pond snail to dishes of saline, and then measured the relative speed by recording the waves produced in the water.
After exposing L. stagnalis' feet to various concentrations of serotonin and cyanide, researchers found that serotonin increases locomotor activity of aquatic gastropods. This can be seen in Figure 1. As serotonin concentrations increased, researchers observed a direct increase in speed. Exposing the foot soles of the L. stagnalis to varying concentrations of cyanide with and without serotonin, as shown in Figure 2, revealed that cyanide brings the elevated frequency of ciliary movement back to basal level. After the L. stagnalis was exposed to aerobic and anaerobic environments, researchers found that smooth muscle cells accounted for a majority, 80% of all soul locomotion in aerobic conditions. This can be seen in Figure 3. And while cilia only accounted for a small percentage, 20% of soul locomotion, they could function successfully with or without oxygen.
Scientists found that in aerobic conditions, 5-HT stimulated locomotor activity and speed of the L. stagnalis, while KCN effectively canceled out the effects of 5-HT by reducing the speed to that of a control. Neither 5-HT nor KCN affected ciliary beat frequency. In anaerobic conditions, 5-HT had little to no effect on the speed of the foot's locomotion. Scientists found that in anaerobic conditions, smooth muscles account for approximately 80% of all locomotor movement, while ciliary beating only accounts for approximately 20%. Researchers concluded that similar behavior patterns would likely be observed in terrestrial gastropods, such as the H. aspera, due to their morphological and functional similarity to those of L. stagnalis, an aquatic gastropod.
This study helped predict the behavioral responses of mollusks in the presence of various stressors, aka the relative levels of serotonin and cyanide, as well as how changes in oxygen and carbon dioxide concentrations in the water as a result of climate change affect mollusk populations.